Thanks very much, Nathan, and thanks to Lee and the others for the invite today. It's a wonderful event so far. And as Nate mentioned, I'm a Hood Lab former Hood Lab investigator. My history starts around 1995. I was a young computational scientist, newly minted with a degree, looking for a career, kind of a wash in many decisions that could be made. And I had a friend who had just started immunology grad school at UCSF. And I'm not sure if you know this story, Lee. And he said, look, there's a guy making waves. He's coming up from Caltech to UW to start this new department exactly for quantitative scientists like you to get them involved in biology based on this sort of then very futuristic notion that biology and genomics in particular is a life science, or is an informational science, is essentially a computational science. So I at the same time, I had bought this book from B. Dalton Bookseller, which I think has been liquidated by Barnes & Noble like 10 years ago. At the time, it was, I think, the biggest bookseller in the US or something like that. And it was all about genomics and how the Human Genome Project would be sequenced. And Lee had, I think, not one but two chapters in this book. So it was synergy. I had a friend telling me about Lee, and then I read about Lee in this book. So I was like, well, how can I get involved in this? So I simply wrote Lee an email, and I said, Dr. Hood, here's who I am. Can I come do a PhD with you? And I think out of the 100 emails Lee must have had that day, he got right back, and he said, absolutely. You have to come to Seattle immediately and start this program. And so I think everything you're going to see today is, plus 20 some odd years, or 15 years at least on that, but all of those conversations and work that Lee and I did together between circa 1995 and 2001 absolutely is underlying everything I'm going to talk about today. So without further ado, why am I showing you a picture of an iPhone? What does that have to do with biology? And it's because I think if you look at where the convergence of computer science and life sciences are going, it's towards intelligent systems like Siri, to which you can ask questions not just Siri, what's a good sushi restaurant in Seattle around the ISB, or what's the weather in Seattle today? It's gorgeous. But questions of a more biomedical and clinical nature, like you just heard about. So questions like, Siri, my patient cells have the following genetic mutations in, say, PTEM and DAM1, and of course an entire omics profile at multiple layers of omics profiles that I can gather on that patient. What phenotypes should I expect, and in particular, what outcomes and therapies should I expect would be effective? So how do we get there? Well, you might be thinking, or you might notice, this is a particular question in a very broad class of questions known as the genotype-phenotype problem. So the classic difficult problem of genetics. And there's lots of reasons why converting or translating genotype or your genome to phenotype have been cited to be difficult. One that gets a lot of attention, and I'm going to focus on in my talk, is so-called genetic heterogeneity. The idea that just as every genome is different, every disease genome is, of course, different. And although there are some genes, say in complex diseases like cancer, where you do observe recurrently over many patients mutations in those genes, by and large, you then have the hundreds of other genes that are mutated in those patients that are ends of 1 or ends of 2s or 3s, these rare events, some of which at least, now some of them might be noise, but some of which at least you believe to be drivers of that disease. But how in the world are you going to find them given there are no patterns needed by either humans or machines to move forward? And that's really the problem of heterogeneity. Now, I flashed forward too soon because there is at least one major weapon that scientists have long had. There are others, but one major weapon that's making a lot of progress these days on this problem of heterogeneity is to aggregate what are rare events at one layer of information, say in the genome, into higher order biological structures and functions, aka pathways. And so this is one of literally dozens of papers in cancer genomics I could have pulled to illustrate this example. If you're familiar with the Cancer Genome Atlas project of the NCI and NHGRI that launched and has now successfully sequenced at least 500 exomes from most cancer types. In almost all of those cancer papers, you'll see a pathway diagram like this, sometimes multiple pathway diagrams like this. And what they're doing here is just, in a fuzzy way at least, they are dividing pathway space here into 5 columns. So one pathway they're calling RTK RAS PI3K. This is head and neck cancer, if I forgot to say that. And for that reason, we have both HPV negative and positive subtypes. They're distinguishing between here, but that's less important for my points. And, so then you have cell death pathways and immunity pathways and so on and so forth. And the important point here is what are rare events at individual genes, FGFR3, 2% of HPV negative patients, for instance, IGFR1R, 4%. Those aggregate into very common events at the pathway level. So if you now sort of make the statement that all of those are part of the RTK RAS PI3K pathway, now you've caught 62% of tumors. And that's a sizable number to which we can analyze and treat. So the idea that pathways are important aggregators of genetic information is by no means new. It's a very old idea. It's a successful idea. The question then really is twofold in my mind. So one is we can only assume, and this is a very poorly characterized answer and maybe even question, we can only assume that we've covered just a fraction of pathway space. We can talk about how much genomic coverage we now have, and that's not even quite 100%, and one can argue about that, but that's a well-characterized question. What about pathway space? The answer is we just don't know, but very smart people think that it's a fraction. It's 10 to 20% at maximum. So the first question is, how do we mount the right experimental and computational efforts to fully cover, whatever that means, pathway space, at least insofar as it's going to be important for understanding disease? So disease pathways. That's question one. And then question two, assuming we even could get that information, and of course we do already, as you saw in the last talk, have information about disease pathways, structure, and function, how do we begin to use that increasingly complete, but right now incomplete information to translate the genome? And so that's kind of the horizontal axis, whereas you could think of a vertical axis here as question one. So I'm going to give you just one vignette in interest of time today for how we started to do that. And of course, there's lots of work both in our lab and in many other labs that I could use to illustrate this concept. And then get into sort of the future of where we think this can go. So here we're still in cancer genetics in the interest of interpreting the cancer genome. And the context of this story is if you look at where global sequencing projects for cancer are going, like the Cancer Genome Atlas, we're now seeing shifts from sequencing just the coding regions, the exons, so-called the exomes of those tumors, now to complete genomes. So if you look back in this Cancer Genome Atlas database, we now have just shy, and this number is probably now greater than that, but at the time of this study that came out earlier this year, just shy of 1,000 tumor whole genome sequences. And so if it was already a problem to distinguish which mutations in cancer genes were drivers and functional and everything, imagine how much greater of a problem it is now that it's not just the 2% of the genome that's coding you're looking at, but it's the other 98% where, as you might expect, 98% of the somatic mutations fall in cancer. So how do you deal with the much greater signal to noise, or the much lower signal to noise, I should say, issue you have there? And it turns out that you actually, in this study, absolutely needed a middle layer of knowledge captured in some type of cancer pathway or network diagram. And the kind that we used here effectively is based on transcriptional connections between enhancers that have been charted in the genome by a succession of large projects like with names like ENCODE, Phantom, and GTEx, and so on. And the links between those enhancers and the genes, they are not known to regulate in most of these cases, but at least there's a little bit of experimental evidence from these projects that those enhancers and promoters do regulate those genes. So the wiring diagram, or the gene circuit diagram or pathway diagram you get out of these things, this is just a small fraction to show you what I mean, looks like this. So you have genes that are nodes, and then you have enhancers or promoters that are nodes. Here, I think it's just 2 enhancers and 4 genes, and then just the connections between them. It's that simple. That's the information that we're going to use to translate the genome in this case. At the start of this study, we had this sort of set of bespoke methods we had cobbled together for mining all of this information. And it turns out right smack dab in the middle of our study, this paper came out of Doran Lancet's group at the Weizmann Institute called the Gene, or which made available this network they called the GeneHancer network. And they've now done all that work for us in a much more sophisticated way than we had been doing it. So we simply, when that came out, adopted this GeneHancer network. And if you read the abstract of their paper, it charts, or they've been able to, by synthesizing all of these projects that came before them, they've been able to chart about 285,000 candidate enhancers. So again, those are the orange nodes connected to respective genes, and all sum told that covers about 12.4% of the human genome. So we're nowhere near 100% of the human genome, but we're way larger than the 2% of just the coding exons. So that's the information that we brought to bear in the study. We had the 1,000 TCGA whole genomes, and then we had this enhancer network. Oh, and I should say, the other layer of data that was absolutely critical to what I'm going to show you is due to the foresight of people in the Cancer Genome Atlas project. They didn't just collect one layer of information at the genetics level. They also had collected mRNA gene expression among other profiles. And so we had, almost for all of these patients, a matched gene expression profile for the tumor prior to therapy. And so that let us look at associating changes or via mutations in non-coding elements like enhancers through the networks to changes in gene expression in the same tumors. That was the key. And so the workflow looks something like this. So first of all, schematically, here are the documented regulatory elements in that GeneHancer network with respect to just one gene now. And importantly, you have not just the locations, but the putative connections into the genes they likely regulate. So now what we could do is look at for every change we see in a given patient in a nucleotide or larger alteration in a documented regulatory elements, we can now build a model, in this case a statistical model based on regression, to try to associate those changes with gene expression, factoring out or controlling for a bunch of covariates that I'm not going to talk about here, but I'm happy to talk about later if people are interested. So what that does for you is it boosts incredibly your signal to noise so that you go from 35 million non-coding sites initially in these 1,000 genomes to grapple with. If you look through successive steps, mapping these things to enhancers, then to genes whose expression levels associate with changes in those enhancers, just down to shy of 200 loci. And if you're familiar with something called an expression trait quantitative locus, or EQTL, or quantitative trait locus, this is just the somatic version of that in cancer, but it's essentially an EQTL analysis. If you have no idea what I just said, don't worry about it, because I've already explained everything. So it should be fine. So is 200 a big number or a small number? Well, it's certainly a small number with respect to the 35 million sites we started with. It's just 200 now. But I would point out it's 192 larger than people knew about before. One of these things was a positive control. So it turns out that the catalytic subunit of telomerase, or TERT, was well documented in a year or two prior to our study to harbor mutations in its promoter that dial up its expression level in a subset of cancers. Here we're finding 192 other putative situations like that. So are these 193 events then common in people's tumors, or are they super rare? Clearly, if they're too rare, they're kind of an interesting point to make, but not significant for clinical care, certainly. But it turns out that many of these, not all 192, but at least the top 20 or 30, are relatively frequent events in different kinds of tumor types. And so here, down below on the columns, these are the different kinds of cancer that were represented across these 1,000 TCGA whole tumor genomes. TERT is here at the top, so the rows are these different enhancer elements that now we think harbor mutations that lead to downstream gene expression changes in tumors. And so TERT here, just before the colon, we have the region of the genome relative to the transcription start site that's affected, and after the colon, we have the gene. So here you have TERT as a positive control. We also find it's prevalent in 76% of melanoma. That was not a new finding by us. But what I want to point out is you have these other findings that are relatively present or prevalent. So 20% or 18% in melanoma for some of these events, 40-something percent for one of these other events. So these are, at least in some cases, recurrent events in cancer. So the next question is can you validate some of these events functionally? And I'm not going to spend too much time on these points. Just to show you one of these events, we've been able to validate about 3 or 4 of them so far, and we're now collaborating with a professor at Stanford who's trying to validate all of them using a new technology he's invented. But the idea here, just for one, is simply to do something like this. So here's one of our findings related to a gene, disheveled-associated gene DAM1. In its promoter, we found one of these events where 5 or 6 patients here, so it's just 6 out of the 930 patients, had mutations in the promoter of DAM1, but again, the network association gives you the power to find these things. That's what's remarkable about it. And 4 out of these 6 events are at the exact same nucleotide, changing a G to an A, and suggestively, that caused, or at least could cause, I should be careful, loss of an E2F family transcription factor binding site and gain of an ETS family transcription factor binding site. So that was looking pretty good. That then led us back into lab to make the mutant form right here at this particular locus, and to put both the mutant and wild type promoters now not upstream of DAM1, but upstream of a GFP fluorescent reporter construct, and then use fluorescent activated cell sorting to look and see if that mutation actually had an effect over a panel of cell lines. Here I'm just showing you one of those cell lines, of course, where you can see a lot more expression here along the x-axis of this GFP construct when you look at the mutant. Now, is that mutant functionally, or is that upregulation event functional? Well, for that, we collaborated with Stephanie Fraley and Emma Farley, both faculty at UCSD. They know their names are very similar to each other. And what Stephanie Fraley did in this case is she's an expert in these 3D matrigel type models of cell invasion, and she was able to show that when she dials up DAM1, it turns cell migration patterns from something like this into something like this. So now at least you have a plausible connection between mutation in the DAM1 promoter all the way to cell invasion in the tumor. So that's that story. But now what I want to talk about is where we think this kind of pathway analysis really needs to go. And part of that is to generalize pathway analysis a bit. So I've talked about enhancer gene connections and, in the context of whole tumor genome non-coding mutations. But there's lots of omics data we've heard about already in this conference, and there's lots of networks and so on, and lots of patient data sets and so on and so forth. And of the many ways that one could prioritize research to generalize this concept, we were most bothered by the fact that all of these pathway diagrams people draw, just like the one I just drew, are these flat circuit diagrams, okay? So a Pentium chip is absolutely a flat circuit diagram. A cell is absolutely not a flat circuit diagram, one could argue. And in particular, when you say pathway, what you actually mean is a hierarchy of structures and functions in cells and tissues, leading all the way from individual mutational events at the sub-nanometer level in the genome, down to changes in individuals at the meter scale. So you have 9 orders of magnitude of physical scale that you're bridging when you're trying to connect genotype to phenotype, first of all. And second of all, just looking at cell biology, which is sort of 1 nanometer to, say, the 10 micron scale, that's already 6 orders of magnitude that you're sort of collapsing down into one pathway diagram or a gene set or an interactome or whatever you want to call it, that did not seem sufficient to us. And so the question was, moving forward, can we do this kind of thing, this genomic aggregation, but in the context of this hierarchy of cellular and tissue systems? And of course, the hierarchy naturally lends itself to aggregation. You aggregate here individual nucleotides into genes, or at least domains, and then genes and their gene products aggregate into protein complexes and supercomplexes. Those protein complexes and changes that impact their function aggregate further and further and further, and so on. So that was the idea. And I really got confident about this idea when I read this piece, which took me really until about last year to find. And it's absolutely transformational, I think, for our field, and totally ignored. And so it's by this guy, Herb Simon, who is a very established professor at Carnegie Mellon for many, many years. He was awarded the Nobel Prize for Economics, but he's equally appreciated for his ideas in artificial intelligence back in those days. He wrote two very influential pieces, at least, maybe three, one of which was called The Architecture of Complexity. And in this piece, his entire point is it's meaningless to consider complexity unless you have hierarchy. That's the entire point of the article. And I'll simply read from the introduction of this article. This is, I think, paragraph 3. I think that's paragraph 2. So it's at the very beginning. The 4 sections that follow discuss 4 aspects of complexity. The first offers some comments on the frequency with which complexity takes the form of hierarchy. The complex system being composed of subsystems that, in turn, have their own subsystems and so on. The second section theorizes about the relation between the structure of a complex system and the time required for it to emerge through evolutionary processes. Specifically, it argues that hierarchic systems will evolve far more quickly than non-hierarchic systems of comparable size. And there is a well-known example you might have heard of if you haven't even read this article, where he talks about 2 watchmakers, one who builds a watch based on just a flat circuit diagram of the components connecting to each other, another who builds a watch and all of its complex parts based on hierarchical modules. Okay, and makes a pretty convincing argument that you could just never build a watch with the former way. You have to use the latter method to build a watch. Also notice here that a supposed, economist or artificial intelligence scientist is talking about biology. Absolutely, in this article, it's all about biological systems for the most part. So please check it out if you haven't already. And what I'm going to do in my remaining few minutes here is just talk about our first stab at this, at what we think the next generation system that's a more general type of system than I just talked about might look like. It is very preliminary, but I hope that's the point of, I can get away with this in this context, in this audience. And the other thing I'm going to get away with and just admit to immediately is, although the intention is to worry about human health and disease and cancer and those kinds of diseases, here I'm going to retreat to the genetic safety of the budding yeast Saccharomyces cerevisiae. Why? Because I think all of us appreciate that, or many of us appreciate that with any new technology or idea, if you can't do it in yeast, it simply isn't going to happen in any other system. We also have, as you'll see in a second, just a tremendous amount of data with which to explore these kinds of budding ideas in both computationally and experimentally. So what does that system look like? It really has 2 parts. So first, rather than so as we gather these interactomes, these connections among proteins and metabolites and genes, like the enhancer gene network I showed you, of course, there's other kinds of interactomes people are busy gathering, protein-protein physical interaction maps that can eventually become comprehensive. There's a lot of excitement about some of those. Certainly gene-gene correlation maps is something a lot of people are building, and so on and so forth. So lots of ways of measuring whether 2 proteins are close or interacting in some kind of physical or functional space. And all of those ways end up with some kind of network diagram that is shown here really is just a hairball, okay? But the idea should be not to use these at face value, but really that's too close to the data. We should be taking these and using them to infer models and assemble models of hierarchical cell structures and functions. And so the first step is to do that, is to start looking at these interactions really is just distances among biological objects, like pairs of proteins and larger groups of proteins. And just like as you use distances at a sort of atomic level to assemble structures of proteins, now use distances among proteins themselves to assemble structures in cells. It's really that simple, okay, in terms of the concept. You just need the right algorithms to do it and the right data, of course, to do it. So that results, or that's going to result in a hierarchical map of systems in cells, at least, because we're at a single cell level here with budding yeast. And then in phase 2, the idea is to then fit a machine learning system, in this case a deep neural network, as I'll talk about shortly, into that system. But importantly, while a lot of people in machine learning are fitting black box systems that can make very good predictions, or sometimes not so good predictions as the case may be, this is absolutely not a black box because it's rooted to the cell biology you have just captured in that hierarchy. So that's the idea. So how does it look? So again, this is budding yeast, and I'm going to move quickly and high level, but you can check out some of the papers. So back in 2013, we focused first on the structural problem here of how can you build a structural hierarchy of what's in cells, of the systems in cells. And to do that, we applied really 3 major types of gene-gene and protein-protein interactions, starting with protein-protein physical interactions, but then moving to synthetic lethal interactions, co-localization in images, and actually as a fourth, co-expression networks. And if you apply the right suite of algorithms to those network data, one can use the fact that proteins are close to network space to infer a module, but then one can see that's nested inside of a larger module, and finally the cell. So here you can see the hierarchy we get. Here's the root of this thing. It's the cell. Very quickly, the data trifurcate into a membrane compartment, a mitochondrial compartment, and an intracellular compartment. So essentially here, we're finding the mitochondrion as basically a cluster of protein interactions at a very, very large scale. Not the best way to find a mitochondrion, but it's there in the data. Okay? And then, of course, within the cell, you have a further factorization into the very many exquisite components a cell has. And many of these are known already, and many of them are novel. But let me just keep moving. So other than to comment that, in fact, this thing, of course, is quite large, just as there's a lot of cell biology that can be captured in these networks. So in sum, we infer about 3,500 subsystems within the cell, that is to say leaves, and, well, actually all nodes under this root, both the intermediate components and all the way down to the gene components themselves. Those are the leaves. All told, that's about 3,500 subsystems. So now the question is, can we train this and give it function? And that's where this deep learning aspect comes in. So for here, we take that exact hierarchy of about 3,500 cell systems, and now to use this hierarchy to translate how mutations in genes give rise to changes in phenotypes, we first had to have a data set that bears on genotypes and phenotypes. And here is again where yeast is incredibly useful. So essentially, many labs, but the largest chunk of these data came out of Charlie Boone and Brenda Andrews' group in Toronto in this Costanzo 2016 publication. But many labs have contributed to this trove of about 12 million yeast genotypes for which we have, if nothing else, a growth rate measurement for the cells. And so if you think about what these, or if I tell you more about what these genotypes are, they're by and large single gene deletions, double gene deletions, and in some exceptions, 3 triple gene deletions. If you think about how does that get up to 12 million? Well, if you do the math, if there's 5,000 yeast genes, that means there's 5,000 choose 2 or 12.5 million pairs of genes, and there you go. So that's how we have such a large sample size, much larger than we have for patients at this moment, for instance, and cleaner. One could also say less relevant, but certainly cleaner. And so that's what we put in as samples for training our machine learner. We have these 12 million individuals for which we have a growth rate down here. And the idea is to do that learning by assigning neurons or artificial neurons in a neural network to each of these cell components. And we assign, in most cases, not just a single neuron, but a whole bank of neurons. So for instance, let's say this component is DNA repair. Those are the bank of neurons we assign to DNA repair. The children of DNA repair in this hierarchy are the sub-processes of DNA repair, things like double-stranded break repair, base excision repair, mismatch repair, and so on and so forth. And each of those have states represented now by neurons. So importantly, this hierarchy is no longer just functional or structural, it's also now functional. And finally, the last thing I have to tell you is we only allow the state of DNA repair's neurons to update based on the states of that child or set of child systems of DNA repair. So that's how we constrain this neural network biologically. So given that, we then apply all the tricks of deep learning, and I should say that bag of tricks is rapidly evolving. And so it's been a challenge just staying on top of the field, and really it's thanks to one scientist in my lab, Zhenzu Ma, who is essentially joined my lab coming out of a premier machine learning lab, who stays on top of the conferences. Otherwise, you would just fall behind. Things are moving so quickly. So first of all, how well does this thing predict? Well, it predicts pretty well, or I wouldn't be giving this segment of my talk. So here are the predictions of this neural network. Here are the measurements. This is in cross-validation, so you're holding part of the yeast genotypes back and testing the system later with them. What is that as a number? It's about 50% or 0.5 as a correlation, if I just fit a line there. Is 0.5 good or bad? Well, one would think it's better than 0, but it's nowhere close to 1. However, I would point out that if we simply in my own lab, and I think it's true in Charlie and Brenda's labs too, if you simply try to reproduce this prior data set, there's of course a limit of reproducibility, and for us, if we simply go into lab and reproduce experiments, we're at about 0.67 correlation. So we think the practical upper bound that we could hope our machine learning model to do would be about 0.67. So we have some room for improvement, but it's not all that bad. Now, in interest of time, which I'm out of, I will skip over comparisons to prior work and just focus on this one bar over here at the very end related to what happens when we don't constrain the system and just build a straight deep learning system out of the box with no biological context. The answer is you get about the same predictive ability. In fact, it's maybe a decimal or a 0.01 bit higher than our 0.5 correlation here, but it's not significantly different than our model gets, but that's biologically constrained based on this hierarchy. However, that thing you cannot interpret. And in fact, if you look at simply bootstrapping the data or even building, it turns out there's some stochasticity in the process of training a neural network. If you simply retrain and retrain and retrain, you get utterly different network structures that make no biological sense. Ours is the same to a first approximation every time because of that hierarchy constraint. And so here's how you can then look at the hierarchy to interpret interesting genotype-phenotype relationships. So here's an example where we had a pair of genes, PMT1 and IRE1, that when deleted result in a dramatic loss of cell growth. And so here, blue represents the loss of the state. So if a gene, the loss of the state is a gene knockout. At the cell level, a blue color means the cell does not grow nearly as quickly as wild type. But now we can look at what are the neurons in this model that are the most affected by this genotypic perturbation at the input, and they all map to these two processes, the ER or endoplasmic reticulum unfolded protein response and its parent process response to ER stress. And it turns out I'm out of time, but you can then go back into lab and with a HAC1 promoter, which is responsive to the state of ER unfolded protein response, you can validate that state in the lab. So let me summarize, and I'll do it really quickly with 4 bullet points. First, I've tried to argue here, hopefully successfully, that if we're going to understand genomes, we really need to think not just about that layer of data at the nanometer scale, but everything, or mapping some details that happen between the nanometer and the 10 micron scale. Those are the pathways of cell biology, and probably above that into tissues and organs. that's the first point, and probably the most important point. I've given you one vignette showing how we made some progress doing this for the non-coding cancer genome using this very large transcriptional network model put forth by projects like ENCODE and Roadmap over the past 5 years. But then in the last half of my talk, I've tried to generalize and look more futuristically about how can we move into the future in a way that we couple the advances of deep learning and other machine learning tools with basic knowledge of cell and tissue biology, which is at the present time wholly lacking from those efforts. Thank you very much. I've already cited most of the work and the people who did the work, but I thank you for your attention. I'll be happy to take questions shortly, I guess. Thank you.